Hello everyone and welcome to Pocket Size Bio with Dr. Casco. In today's video, we're going to look at part 2 of chapter 10 of Campbell's Biology, which is photosynthesis. Okay, so this is 10.5, alternative mechanisms of carbon fixation have evolved in hot, arid climates. Okay, so let's get to it. As we see the pictures and images here, definitely these adaptations and these mechanisms that we're going to talk about involve the stomata and gas exchange. So that's kind of the primary thing that we're going to focus on. So one of the major problems facing terrestrial plants is dehydration. And we know dehydration, you have a lack of water inside the organism, you're dehydrated. Metabolic adaptations to reduce dehydration often require trade-offs with other metabolic processes, especially photosynthesis. And we're going to see these particular trade-offs in these two sections here, or in this section here. First, let's talk about the stomata. We mentioned this in the chapter on the organelle and the tour of the cell, right? But just to kind of revisit here, the stomata, they're the major kind of structures of gas exchange. So here, in this particular route of gas exchange, you have carbon dioxide coming in and oxygen coming out or going out. Okay, so this is the central structure of a stomata. You have these guard cells that they swell and then they become a little bit flaccid, and depending on their amount of water, they would close this opening or open this stomatal pore. Okay, so this is a diagram showing a stomata. This is how it will look like under a light microscope of a particular leaf here. And at the same time of gas exchange, they're also the main site of evaporative loss of water. Okay, so on hot, dry days, plants generally close their stomata to conserve water. Carbon dioxide concentrations in the leaf airspace, because the leaf itself has some open spaces where you can have gases floating around. So because their stomata is closed, you don't have CO2 coming in. So the CO2 is being consumed by photosynthesis. It's going to start decreasing. And at the same time, oxygen concentrations from the light reaction will increase. Okay, so these conditions within the leaf favor an apparently wasteful process that we are going to talk about called photorespiration. Okay, photorespiration. So scientists refer to this as an evolutionary relic, okay, or evolutionary baggage. And what happens here, generally, in most plants, what are called C3 plants, they, the initial fixation of carbon dioxide comes in and will be, consumed or used up with RUBP using Rubisco, okay, and that will produce a 3-carbon compound called 3-phosphoglycerate. Okay, 3-phosphoglycerate, and then that will continue inside the Calvin cycle and whatnot. So these C3 plants, they include rice and wheat and soybeans, what we commonly think about. Now, the thing is, when stomata and C3 plants partially close on hot, dry days, as I mentioned before, carbon dioxide levels drop. And the reason why, again, and this is kind of the main thing that you need to understand, carbon dioxide, the play of carbon dioxide within these environments, right? It's being consumed in the Calvin cycle. Okay, so it's being consumed in the Calvin cycle, At the same time, okay, oxygen levels will begin to rise. So you're starting to get an increase in oxygen levels because you're having the light reactions. Remember the light reactions using sunlight, right, to kind of channel electrons, split the water into electrons and hydrogen and so forth, and oxygen is being produced. So oxygen levels are rising. Although Rubisco normally accepts carbon dioxide in the usual Calvin cycle, what happens is that carbon dioxide becomes so scarce that Rubisco will start accepting oxygen to RUBP. So you see here on this left side in red, you have oxygen and RUBP, ribulose biphosphate, will come in and be catalyzed, right, using Rubisco, and it'll start creating, two compounds, right? A two carbon compound and a 3-carbon compound, what is called glycolate. Now, this might be a little bit beyond the scope of even Campbell biology, but I kind of put it here for you guys to see here. Now, that process where you're using photosynthesis and applying photosynthesis and using the oxygen from the light reactions to respire, consuming oxygen now, is called photorespiration. This 2-carbon compound is exported from the chloroplast and then degraded to carbon dioxide by mitochondria and peroxisomes and so forth. So you're getting oxygen in and you're producing carbon dioxide. Okay? And you're producing carbon dioxide. Now, as if you're seeing here in the image, okay, what's happening? You're getting these these different compounds. But here's where I want you to see photorespiration actually consumes ATP. That's one of the key things you'd also need to note. So they use oxygen and they consume ATP. This process doesn't produce, it consumes ATP. So photorespiration also doesn't produce sugar. Okay? Photorespiration does not produce sugar. So ultimately, quite interesting that the photosynthetic output by this process actually is kind of a consumer. It's siphoning organic material from the Calvin cycle. It does release carbon dioxide, but it also consumes ATP. Okay? It also consumes ATP. All right. So again, siphons organic material from the Calvin cycle. So it's kind of removing material from the Calvin cycle, and it releases carbon dioxide. So scientists think that the existence of photorespiration could be an evolutionary relic or baggage where it was once used, where and there as a kind of a standby process. When Rubisco first evolved, the atmosphere had far less oxygen, more carbon dioxide than it does today. Nowadays, there's more, you have a lot more oxygen there. So there was probably an active site of Rubisco to kind of, manipulate oxygen. So in a way that it could be used. So that in that sense, you have kind of this secondary process where it isn't as very effective anymore. Okay, so let me just go to this next part here. So in many plants now, including crop plants, photorespiration drains away as much as 50% of the carbon fixed by the Calvin cycle. So it's a quite consumer in that sense. So at the same time, photorespiration could play a protective role in plants. Okay, plants are generally defective in their ability to carry out photorespiration, are more susceptible to damage induced by excess light, breaking down, let's say, like oxygen, maybe into a raw species. So photorespiration can act to neutralize damaging products of the light reactions. So you want to manage that also, right? No excess electrons and oxygen there that can potentially be turned into some ROS products, reactive oxygen species products. Okay, so again, these kind of damaging products could build up when low carbon dioxide concentrations limit the Calvin cycle, so the Calvin cycle can't forward, right? It can't continue. So heterotrophs are dependent on carbon fixation in chloroplasts for food. Humans, and this is quite interesting, we view naturally we view photorespiration as wasteful because we want chloroplasts to be producing sugars as part of the Calvin cycle. If photorespiration could be reduced in certain plant species without otherwise affecting photosynthetic productivity, crop yields and food supplies might increase. Again, I want to note that if photorespiration could be reduced in certain plant species, it's possible that we could be using the carbon and joining more the normal Calvin cycle to be producing what we need to produce, right? Food supplies, right, might increase. Certain species have evolved alternative modes of carbon fixation to minimize photorespiration, and this is the next kind of process that we need to take a look at in C4 plants. So in one strategy, these type of plants, they fix carbon dioxide in a 4-carbon compound, thus being called C4 plants. No, not the explosion. Okay, not the dangerous weapon you hear about. So yes, many plant families belong to this group of plants, right? Corn, sugarcane, grasses can fix carbon dioxide under certain conditions. Again, let's take a look at the stomata. We're gonna talk a little bit more about the leaf and some of its kind of structures here. So partial closing of stomata. So in these plants, the stomata gets partially closed, so water can get conserved. You don't want to waste water, especially in places that are very arid, okay? Or it's just very dry, or there's a little amount of water in the area. So you want to conserve water in hot and dry days. Again, because you want to conserve water, stomata gets partially closed, and at the same time, as a consequence, carbon dioxide gets reduced. Sugar can still be made in these particular plants. Okay, so there's a unique leaf anatomy within these particular plants that correlate, with a particular mechanism of C4 photosynthesis. And there's 2 structures that you do need to know. You have your mesophyll cells that you probably remember from, again, the chloroplast and the leaf, talking about plant cells, and also bundle sheath cells. Okay, and just to show you their location, so you have a leaf, kind of cross section here, and you have vascular tissue, the veins where water is moving, up and down in one direction. So you have this vascular tissue. Surrounding this vascular tissue are these thick cells, right? These are the bundle sheath cells that are surrounding this vascular tissue. And then in the surrounding area, you have your mesophyll layer, okay? These individual cells that they have some spaces in between them and so forth. And then if you were to look at them more in detail, so they're connected, okay, by these little channels called plasmodesmata. In any case, you have mesophyll cells. They're kind of anchored together with bundle sheath, and something is occurring between them. So let's take a look what these cells are to begin on. So bundle sheath, the name tells you they're surrounding the veins, right? This vascular tissue. And within the leaf, the Calvin cycle is confined to the chloroplast of the bundle sheath cells. So Calvin cycle is occurring here in the bundle sheath. Okay, they're tightly packed. And then you have the mesophils, as I mentioned before. They're more loosely arranged between the bundle sheath and the leaf surface, the epidermis there. Okay, so one thing we need to be critical is this following part here, highlighted in black and yellow. The Calvin cycle is preceded by the incorporation of carbon dioxide into organic molecules of the mesophil. So what's happening in particular? Let me just put some space so you can distinguish there. Into the mesophil. Okay, so what happens? You have a key enzyme called phosphoenol pyruvate carboxylase. So the phosphoenol pyruvate could be short for PEP. Okay, and this is the enzyme called carboxylase. So you could call this PEP carboxylase or PEP carboxylase. So what this does is that this enzyme adds carbon dioxide to phosphoenol pyruvate. And this forms the 4-carbon product called oxaloacetate. Remember, this is occurring in the mesophil. So just to kind of zoom in here really quick, you have carbon dioxide coming in. Okay, it is joining this PEP, this phosphoenol pyruvate. They come in together, get interact with this carboxylase, and they turn into oxaloacetate. This oxaloacetate will move into, again, it does undergo certain transformation into malate. It goes inside now the bundle sheath cell, okay, through these channels, the plasmodesma. Okay. And now we go to the next step. All right. So this carboxylate has a high affinity for carbon dioxide. Just a little bit of more detail here. What is important is that this enzyme, yes, has high affinity for carbon dioxide and no affinities for oxygen. So what does that mean? You don't have to worry about photorespiration occurring because this is a very carbon dioxide specific situation. Okay, so PEP carboxylase can fix carbon dioxide efficiently when Rubisco cannot. Remember, Rubisco is fixing oxygen in photorespiration. In this case, you have another enzyme, so make sure be mindful of that. Okay. Just to continue on with the notes for you guys here, and I've re-added this image here so we don't lose it. Okay, so again, as I mentioned before, they're transported into the bundle sheath cells. Okay, and what happens is that this oxaloacetate come in, convert into malate. The carbon dioxide then gets removed from this 4-carbon compound. Okay, so now we're here. Carbon dioxide gets removed from this 4-carbon compound, and then you get pyruvate again. This pyruvate, okay, goes back into mesophil cells, and with ATP gets then kind of reestablish into phosphoenol pyruvate or PEP for the cycle to continue again and again and again. So one of the things you also need to remember is that ATP is being consumed. Okay. And then you want to keep doing that and keep doing that and keep doing that so you can produce carbon dioxide. Okay. And to generate more ATP, what cycle, if you remember the cyclic flows of the electron transport chain, we have linear and cyclic. In these particular cases, you have the cyclic electron flow occurring because you want ATP to be generating and generating, if you remember that part, right? So these cells tend to contain photosystem 1 and not photosystem 2. So you have cyclic electron flow. Okay, you produce more ATP, you can enable this pathway to be continuing. Okay, So again, mesophyll cells dump carbon dioxide into the bundle sheaths. This keeps carbon dioxide levels high, so rubisco can keep accepting carbon dioxide. And this is a very nice kind of key sentence here for you guys to get the main idea. Let me just highlight that here. Carbon dioxide levels need to be high, right, to maintain carbon dioxide into the Calvin cycle and not oxygen. Okay, and this is what we see here. Ultimately, to produce sugar, that sugar could be passed down to the phloem of the plant. Okay, so this can be thought of as a CO2 concentrating pump powered by ATP. Okay. So C4 plant photosynthesis decreases photorespiration and increases sugar production. So it's a very fail-safe kind of mechanism to prevent photorespiration from occurring. Okay. C4 plants generally thrive in hot regions with intense sunlight. Okay. Since the Industrial Revolution, just a little bit, of additional information on human impact. Yes, human activities have been burning. We know this. We're familiar with this idea. Human impact burning fossil fuels. We're providing an increase in carbon dioxide, climate change, and increase in temperature has been affecting even these metabolic pathways. There's been an increase in carbon dioxide concentration and temperature. And depending on the plant type that you're observing or that exists, these situations are being affected differently from the environment. So there could be a change in relative abundance of the plant species just because of climate change and an increase in carbon dioxide levels. Okay, so yeah, which type of plant? A good question would be, which type of plant would stand to gain more from increasing carbon dioxide levels, right? C3 plants, the binding of oxygen rather than carbon dioxide by rubisco would lead to photorespiration. So therefore, there would be a lowering of photosynthetic efficiency. C4 plants would concentrate carbon dioxide in bundle sheath cells at the cost of ATP. So it's quite interesting what's happening there. Remember, we're talking about not just carbon dioxide levels, but also temperature and heat, where you have to close the stomata, right? So it's a problem, right? Rising temperatures can have the opposite effect, to increase photorespiration. Okay. Ultimately, amongst these two, C4 plants would largely be unaffected by increasing carbon dioxide levels. Of course, they want carbon dioxide levels in the situation, in the environment. Okay. In different regions, a particular combination of these two factors is likely to alter the balance in population or relative abundance. So this is something that ecologists and botanists need to be on the lookout to see how plant diversity is changing based on global conditions. Okay. And then another type of adaptation, a second type of actual adaptation in arid conditions. Let me just zoom in here. Has evolved in succulent plants, cacti, pineapples, and other of these particular plant families, right? And what happens here is that these plants, they open their stomata at night and close them during the day. Again, you want to conserve water. Okay, you want to conserve water, but at the same time, you're preventing carbon dioxide from entering the system during the day. Okay. Let's say you have very, very dry savanna, desert type regions where it's just so hot during the daytime. During the nighttime, though, temperatures do lower, humidity is higher. So these plants fix carbon dioxide into a variety of organic acids. Okay, organic acids um, that they can process later. And they produce these organic acids in the mesophyll cells. This method of carbon fixation, okay, is called crassulacean acid metabolism. Okay, if I'm saying that right, crassulacean acid metabolism, or for short, CAM plants or CAM plants. Okay, C acid metabolism, crassulacean acid metabolism, or CAM plants. So what happens here is that these cells, they store organic acids that they're making during the night in their vacuoles. So their vacuoles can serve as a kind of backpack to house all these acids that are containing these carbon atoms. In the morning, when the stomatas close, they will liberate these organic acids where they could be used into the Calvin cycle. They can extract the carbon dioxide so they could be incorporated into sugar, right? So here's a particular diagram. You can see here a C4 plant, sugarcane, versus a CAM plant like pineapple. Same situation here, where you have mesophyll cells. They're undergoing this type. And this is again C4 plants. They undergo this organic acid. Remember oxaloacetate to collect carbon dioxide, and then they will liberate that carbon dioxide in bundle sheath cells. Calvin cycle produces sugar. Okay. In these CAM plants, what differs in this particular case is day and night cycling. Okay, during the nighttime, they'll harness the organic acids, the carbon dioxide, into these organic acids. And then during the daytime, they would remove this carbon dioxide into the Calvin cycle to produce the sugars. Okay, to produce the sugars. So again, they add carbon dioxide, both, C4 plants, CAM plants, they add carbon dioxide into organic intermediates before they enter the Calvin cycle. C4 plants, carbon dioxide and carbon fixation is separated between these two types of cells. But in CAM plants, carbon fixation and the Calvin cycle are separated temporarily, so day and night. Okay, so both of these types of plants ultimately will create sugar out of carbon dioxide, one way or another. Okay, just give you these concept checks. All right, so this is a very nice comparison of these two kind of pathways here. So with that, we finish the alternate methods, right, of carbon fixing. Photorespiration, you have C3 plants, photorespiration and C3 plants, C4 plants, and CAM plant metabolism. Just to kind of summarize there. The last section of this chapter, photosynthesis is essential for life on Earth. This is just an overview of how the importance of photosynthesis in itself, and the main reactions that occur. The light reactions ultimately capture solar energy, use it to make ATP, transfer electrons, and form from water to NAD plus to produce NADPH. Calvin cycle uses ATP and NADPH to produce sugar from carbon dioxide. Okay. Ultimately, all that energy from sunlight is harnessed from chloroplasts, so it can go from light energy to chemical energy that is found in organic compounds. Okay. Sugar can be made in chloroplasts that ultimately can supply the entire plant with chemical energy, and also as a backbone for carbon skeletons to produce different types of organic molecules depending on what the plant needs. In respect to photorespiration, about 50% of organic material is consumed as fuel for cellular respiration in plant mitochondria. Some photosynthetic products are lost as photorespiration. So remember that, as I talked about earlier. Carbohydrates in the form of disaccharides, more specifically sucrose, travels in the veins, right, in the phloems, to non-photosynthetic cells in plant bodies. Not every single part of the plant can photosynthesize. Of course, you need nutrients on certain areas of the plant, like the root, for example. Okay. Sucrose provides fuel for respiration, the raw materials for anabolic pathways, including synthesis of proteins and lipids and cellulose. Okay, cellulose, as we've seen before, very, very simple here. They're the main ingredient for the cell walls that helps provide rigidity and structure for the cell plant, support for the cell plant, for the plant cell plant. Sorry about that. Is the most abundant organic molecule in the plant and probably on the surface of Earth. Okay, very, very important. I'm going to show this panel for you guys so you can take a look. This is a very nice summary for you guys to explain to yourselves the overall process of photosynthesis, what's happening on the outside of the plant at the macroscopic level, and then within the plant cell, more specifically the chloroplast. Okay. So lastly, plants also store sugar or excess sugar as starch, right? Starch can be stored in chloroplasts and on roots, tubers, seeds, and fruits. Heterotrophs, including humans, right? We completely or partially consume plants as a source of nutrients, right? Of fuel or raw materials. On a global scale, right? So this section is kind of providing an insight on how important photosynthesis is, right? It's the most important process on Earth. It is responsible for the presence of oxygen in our atmosphere. Each year, photosynthesis synthesizes 160 billion metric tons of carbohydrate. It's kind of mind-blowing. No process is more important than photosynthesis to the welfare of life on Earth. Yeah. Pretty cool. Let's take a look. So just to go over the chapter review for you guys to see. Just to skim through down there for a bit. All right. This is just the second page of the chapter review. Sorry about this throat noise. All right, guys, so that's chapter 10 on photosynthesis. I hope this video was helpful for you, and I'll see you in the next chapter. Thank you very much for watching, and take care.